Dr Rhoda Hawkins, who's going to be giving our next talk. Rhoda is in the Department of Physics and Astronomy in the University of Sheffield. Her background is in physics. She did her first degree in physics in Oxford, and then a PhD in polymers and complex fluids in the University of Leeds, and has moved around in a number of places since, with appointments in Amsterdam and Paris. And she's also a trustee of Christians in Science, and she applies her physics to biological problems. So she's going to be talking to us today on order, disorder in biology. Rhoda.
Well, thank you for that introduction. Am I on? Yes. Thank you for that introduction, and thank you for the invitation to come and talk to you this morning. I have not spoken at a Faraday course before, but I have been a participant before, so it's quite exciting for me to come and talk to you this morning. My title is slightly intriguing, really, Order, Disorder in Biology, and I hope as we go through the talk you will begin to understand what I'm meaning by that title. As you've heard from the introduction, I'm a physicist by background, but before any of you start panicking, I have made sure there is not a single mathematical equation on my slides. I will be referring to the fact that I do mathematics. I'm a theoretical physicist, and therefore mathematics is the language in which I work for my research. But I'm not going to inflict mathematics on you on a Saturday morning. So, rest assured, we're going to go through, I'm going to talk about some of the results of the things that use mathematics, but I'm not going to expect you to follow mathematics during this presentation. But because I come from a theoretical physics background, I come at questions in biology maybe from a slightly different perspective from some of the other speakers, and that hopefully will be interesting for you. So let me let you know what I'm going to talk to you about this morning. The plan of what I'm going to talk about is I'm going to give you an example of something which I see as an ordered and in some ways purposeful system in biology. The example I'm going to use is that of cell migration. That's cells that are moving. And then I'm going to delve into more microscopic details of that system. We're going to look inside that cell at the component molecules and their dynamics and their interactions. And I'm going to show you how many of the things that are happening at that more microscopic level appear more disordered. And so we've got the order, and then we've got the disorder. And then I'm going to return to the larger scale behaviour and talk about some interesting properties, and this is where some of the physics comes in and where it touches on my current research. And I'm going to finish with some comments on the slightly more theological perspective, talking about emergence and beauty and wonder. So I want to start by showing you this little film clip. It's a classic film, and what you're seeing here is a white blood cell that is moving. And it's chasing this little black thing, which is a bacterium. Now, the other things in the way are red blood cells. They're not so interesting for us, apart from the fact that the white blood cell has to push them out. Ah, he's got it! Did you get that? It got it. In the end, it got it. But it was chasing the bacterium, and in the end, it got the bacterium and engulfed it. Now, you could see that as a bit of a fight between the white blood cell and the bacterium, and in this case, the white blood cell won, which is pretty good, because that's what keeps us healthy. Our white blood cells are part of our immune system, and they are chasing infections such as bacteria in order to keep us healthy. Now, I look at that little film clip and I love it. I've seen it so many times before, and yet every time I see it, it just makes me smile, as it made many of you smile. I will never tire of that little film clip, because there's so many elements of it, and there's so many questions it raises for me. And as a physicist, I'm interested in the material properties of things. And I look at these cells, and, well, they're just blobs of squishy things. And yet it's clearly so much more than a blob of squishy things, because it's doing things that are particularly interesting. It's moving around. It appears to have purpose in the sense that it is following the bacterium, chasing it. So it seems to have got a particular direction. and it seems to be acting as an organism doing a job. And so I'm interested in my research in trying to understand systems like this from a physics perspective, and working alongside biologists, working together to see if we can understand better the mechanisms of how something like that works. So that's the overall picture and the motivation of where I'm going. But let's start to understand this system by delving a bit deeper. So what I'm going to do is I'm going to do something that is classic within physics, has been classic within physics for a long time, and more recently is also classic in biology, and that is a reductionist approach. I'm going to start with this overall system, and I'm going to break it down into constituent parts and talk about those bits and see what we can learn from the overall, see what we can learn about the overall behaviour from the bits inside. So this is a reductionist approach, and then I'm going to talk later about what that can teach us. So first of all, let's look inside this cell. So this is a picture of a cell, and it's labelled in a particular way. Please ignore these bits around there. We want to focus on this one here. This is a cell, and there's things that are labelled. First of all, the blue bit here in the middle, that's a stain that's labelling the DNA. And we've had a fantastic talk this morning about the DNA, and I'm not going to be talking about DNA, but I've thoroughly enjoyed this morning, and so I wanted to point out to you, that's the DNA in the middle, in the nucleus. This is a eukaryotic cell, not a bacterium, so it's got a nucleus. And the other things that are labelled here are the green things. These are protein filaments that we call microtubules. And around the edge, the red bits are another kind of protein filaments that we call actin. I'm not going to throw too many protein names at you because as a physicist I know how off-putting that is when you're faced with hundreds of names and they're all complicated. But there are two names here I'm giving you at this stage, microtubules and actin. And these together, these two protein filaments, the green ones and the red ones, microtubules and actin, they make up what we call the cell's cytoskeleton. Now, the word cytoskeleton has the word skeleton in it, and that is analogous to our bone skeleton, and cyto just means cell. So this is like the skeleton of a cell. It's like the skeleton of a cell in a few different ways, in the sense that this structure of proteins is what gives the cells their shape, and it's also involved intricately in cell deformation, so changing shapes, and cell movement. So in that sense, our skeleton gives us our overall shape and some of our overall physical properties, and we have to move our bones around in order to walk. So there's some analogies there, and that's why it's helpful to think about it as a kind of skeleton. But our bones are very rigid, they're solid, and these filaments are far from that. So let's delve a little bit deeper into what these things look like. So first of all, I'm going to show you a picture here of actin. This is right at the front of the cell, right at the edge. Somewhere like this, we're blowing that little bit up, and we're looking here at what that looks like. So this here is the edge of the cell, and then you see this crisscrossing network of these long filaments that are, in this case, are actin. Now this image here is taken with an electron microscope, and that's a fantastic technique giving high resolution. However, there are issues with it in the sense that, for example, I'm telling you that this is actin, but it's not labelled. So we're assuming it's actin, but there's probably other stuff in there as well. But we can't tell the difference between the actin and the other stuff because there's no specific labelling here. But this is really high resolution, showing us this crisscrossing, complicated looking structure. Now, I'd like you to see that it looks pretty disordered. It's very branched, there's filaments all on top of each other, all over the place, and so it looks quite disordered, and that's important here. So I'm now going to show you another picture of, so this is using electron microscopy. Now using light, this is also of actin at the front of the cell, but this is using light microscopy, and this is using a new technique within super resolution microscopy. I don't know if some of you may be aware that over the past 10 years or so there's been a revolution in light microscopy, and we can now beat the diffraction limit and see things smaller than we ever could before using techniques from super resolution. This is a technique that's called STORM. It's being used to make this image. And you see these similar branched structures, which is beautiful here. And here, the difference between this and the previous image I showed you is that here we really do know that what we're seeing is actin, because there's a label specifically labelling the actin. So what we see the fluorescence in, we know is actin. And you see this beautiful crisscrossing network, which actually in this looks really quite disordered in some ways. But there's also some order in it as well, with some structure in it. You see here we've got bundles, and we've got some bundles here pointing out. And as I will talk to you more about, there's more order in this than it looks initially. So I want to show you an example of what an actin filament looks like. And this again is an analogy, but it's a polymer of proteins. And a polymer is basically like a long stringy molecule. So it's long and thin. This is actually a piece of washing line. And it's, as we heard this morning, proteins can be thought of as linear molecules, but actually they all curl up. And what we have here is actually a polymer of proteins. So each monomer itself is a protein. I'll talk more about that later. But I'd like you just to have that picture in your minds of this stringy-like molecule. And what we're seeing here in this network is many of these filaments all jumbled up together, and that's why we see this network that looks, in some sense, is quite disordered. Now, I've talked to you about the actin. That's the red bit that I showed you on the first slide. But there was also these green ones, these other filaments, and they're called microtubules. So this is an electron microscope picture of microtubules. You see they're very long and straight, and they're actually tubes. Now you can even see that a little bit on these on this picture. You can see that they're tube-like. We know they're tubes because actually I haven't got the picture here, but if you turn this round and look end on, then you can see little circles. So we know that they're tubes. They're called microtubules, although I think if they had been discovered today, they would be called nanotubules because that sounds more cool. Their diameter is around 25 nanometers, so you could call them nanotubes, but they're originally called microtubes. And like the actin, they are made up of many subunits of proteins called, in this case, tubulin. So these are microtubules, and this is an electron micrograph of a few of them, but I now have a picture of them in a cell. This is by fluorescence microscopy, and this is actually a plant cell. The previous cells I showed you were animal cells. This one's a plant cell, and what I'd like you to see here is that there's this beautiful order of these things lining up. And so there's this beautiful ordered pattern. It's not totally perfect, but if you look at it on average, you've got all these microtubules pointing in roughly the same direction. Now, this is a plant cell. Plant cells are roughly cylindrical. You will find that because I'm a physicist, I see things like circles and spheres and cylinders everywhere because they're so simple and beautiful, and the maths is easy. As soon as you get complicated shapes, then the maths becomes rapidly very difficult. So for me, this is a cylinder. The interesting thing about these microtubules is that they are wrapping around the curved surface of the cylinder. So the axis of the cylinder is this way, and they're wrapping around like this. It's obviously a 2D image, but in 3D they're going around like that. Now, as a physicist, I look at something like that, and I see this order. And in physics, we have ways of describing order of things that are long and thin. If we have a material that has got molecules in it that are rod-like, then we have ways of describing that. And the kinds of materials I'm thinking of are liquid crystals. So this is a cartoon here showing you liquid crystals. And I want to introduce what liquid crystals are. So if you imagine a solid, you know that a solid has got a regular array. All the atoms are in a nice regular order in terms of their positions. And if we have molecules that are not point-like, they're not little balls, little spheres, but they're actually little rods, like I've drawn here, or like a pen here. So we have molecules that look like pens, and in solid form they're all lined up, their positions are all ordered, and their directions are all ordered, they're all pointing in the same way. That's a crystalline solid form. But you can also imagine these rod-like molecules having disordered positions. So their positions are all over the place, but they're all pointing the same way. And that is something we call a nematic liquid crystal. And that's different from what we would call a liquid, where all the positions are disordered, but also all the orientations are disordered. They're pointing in all different directions. So this is like a liquid, this is like a solid, but these kind of materials form something that's in between. We call a liquid crystal, because it's a bit like a liquid in the sense that the positions are all over the place, disordered, but it's a bit like a crystal because there's a certain element of order in the system, in the fact that all of these molecules are pointing in the same way. I've got here another picture. This is of what we call a polar liquid crystal. This is similar to this one, except you see now there's arrows. That's like my pen here, where the top of the pen is different from the bottom. And so therefore, if I've got the pens, a pen this way up, it's different from having the pen that way up. And so in that case, we call it a polar liquid crystal. So some molecules are just long and thin, and other molecules are like little arrows in the sense that one end is different from the other end. And so we need to think about that differently, because you can imagine that that is now different from if half of them are pointing up and half of them are pointing down. So that is a brief introduction to liquid crystals. We use liquid crystals in technology. In fact, all of us are, I expect all of us here, are owners of liquid crystals in our liquid crystal displays, in our gadgets that we seem to be attached to these days, in our smartphones, laptops, etc. But I'd like to draw an analogy between these liquid crystals and this beautiful array of the microtubules I've got here, where they're all ordered. Now, there are differences between the system, but there are similarities here in the sense that we've got this order. So we can use something of what we know from liquid crystals to describe the order in the system. Now here is a plant cell, and what I'm going to show you here is a little video of the plant cell going through its life cycle. It has started. Okay, so now it's pretty disordered, and these are microtubules we see in green, and over time you'll begin to see some order arising. You'll begin to see some stripes arising. You see some stripes arising now from what was initially a very disordered system. We're now beginning to see these stripes arising, and this is happening over time in the plant cells, and it will end up looking like the ordered picture I showed you before. There we go. So we've now got these stripes. Now, these stripes and this order is important for plant cells because the direction of these things dictates the direction of the fibres in the cell wall and helps to build up the mechanical strength of plant cells. But what I wanted to focus on here is this order that's arising from what looks more disordered. So this I've been showing you here at the level of these filaments, but let's go a level lower in length scale and talk about one filament and what's happening to one filament. And one filament here is actually made up of lots of single units. So this is a polymer. A polymer is a long, thin molecule that's made up of lots of what we call monomers, so little subunits. But each of these subunits here is actually a protein in itself. And we can think of a protein as made up of lots of subunits, the amino acids, as we were hearing this morning. So in that sense, this individual circle here is already made up of lots of subunits, but then it makes one unit, which then makes up the larger hierarchical structure here. So here we're focusing on this structure here where we've got all these monomers, these subunits, and they can grow or shrink in the sense that more subunits can add on or fall off. So it's a little bit like this. So here I have little beads for each of the subunits of my filaments, and the end ones can come off and come on like that. And in order to bind on, it actually requires biochemical energy. There's a molecule in the cell that we call ATP, and you can think of it as the fuel of the cell. There's other similar molecules like GTP, and the chemical reaction of hydrolysis of these molecules, ATP going to ADP, is necessary for this to be able to bind on. And that's giving this biochemical energy in order to grow longer with more subunits going on. So these subunits can come on, off. And this is happening, this dynamics are coming on and off, it's happening all the time in these cells. And what you saw in that video I just showed you of the green microtubules being all disordered and then becoming ordered, the way the dynamics of that is working is that actually filaments are continually falling apart and coming together again, adding up, and that's how the dynamics is working in the system, which is very different from the dynamics in a liquid crystal, whereby a liquid crystal normally does not have molecules that can grow or shrink. They're just fixed lengths, and they can change direction and move around, but they cannot grow or shrink. Whereas these biological filaments, they're continually growing and shrinking, and so they're dynamic in that way. Now, I mentioned the energy. I mentioned the energy for a reason. It requires biochemical energy to get more subunits bound onto the end of this. And that can be used to generate forces. So here I have a cartoon of a filament that is growing towards a barrier. So this is a barrier, for example, the edge of the cell, the membrane at the front of the cell. So we've got a filament that's growing towards this barrier, the front of the cell, and more subunits can add onto the end. More subunits can add onto the end as long as there's a gap here that's big enough for a subunit to add on. But if there's not a gap big enough for the subunit to add on, if the barrier fluctuates, moves slightly, then and opens up a little gap, then another subunit can come and bind in the gap. And if another subunit is bound in the gap, then this barrier cannot move back again. So it can move forwards, but it can't move backwards. And so that is generating a force pushing this barrier and pushing it forwards. And here, there's a force here, F, showing the force that this filament is able to overcome a force here, pushing the barrier and push it backwards. So this filament, a filament like this, is able to push the front of a cell forwards. Now you'll see why I keep talking about the front of the cell, because I'm already determining direction, front and back. I'll talk a little bit about determining direction later. But you have going forwards, pushing this thing forward. So these filaments are able to generate a force. Now, for any of you who know any physics, you need energy in order to generate forces. And that's why that biochemical energy I mentioned of it requires energy to add a subunit on, why that's actually crucial here, because that's the energy that you need in order to push, in order to create this force. So that's one way in which these filaments are very different from physical synthetic materials that are non-living, like the liquid crystals in our liquid crystal displays in our smartphones. These are different because they are able to grow and consume biochemical energy and exert forces, and that makes them what we in physics call out of equilibrium, or in my subfield of biological physics, we use the word active. We say that this is an active system. What we mean by that is it's consuming energy in order to do things like exert mechanical forces. I'd like to show you another example of an active system involving these filaments, and that's something called molecular motors. Now, molecular motors are proteins. They are very cool proteins in the sense that they act like little motors. So here is an example of a molecular motor. and I have my filament, and the molecular motor is able to bind to the filament and walk along it. Now, actually, its feet are in a field called heads, which is slightly amusing because, yeah, they're called heads, but they're called heads. They were called heads before we knew that they were moving like this, like feet. So we're going to call them heads because that's the way the history of science works, isn't it? But these things actually bind and they move along. And the way that they're moving along is by binding and unbinding, changing shape slightly and moving along. But to do that, they require energy. So in order to bind and change shape and move along, they require this energy, the same energy molecule I mentioned before, ATP, hydrolyzing to ADP. They require that energy in order to move along. And I talked about them walking along a filament, but they can also bind to more than one filament at the same time. So here, this model is a model of a single molecular motor, but lots of these molecular motors can cluster together and bind to more than one filament at the same time. So this cartoon is attempting to describe that. I've got a filament here and a second filament here, and then a bunch of molecular motors here, such that molecular motors are bound to both filaments at once. Now, if they're bound to both filaments at once, that means they can walk on two filaments at once. Now, molecular motors have a preferential direction. They will only walk in one direction along the filament. So these filaments are like my little arrows that I showed you on my polar liquid crystal, but actually one end is different from the other end. So if you imagine that both of these filaments are pointing in the same way, so their arrowheads are both here, so then this molecular motor can walk along both of them at the same time. But if these filaments are anti-parallel, so I think the best way to show that is with my arms. If my arms are parallel, then the molecular motor can walk along both my arms at the same time together. But if my arms are anti-parallel, then I have my molecular motor here, and it wants to walk in an opposite direction on each filament. Now that is going to cause a stress in the system. And in the case of actin filaments and molecular motors that we call myosin, this causes a contraction. So what happens is my arms are like that, and then they go like that. So they contract. So without molecular motors, start like that, and then they go like that. And the overall system is a contraction. This is actually what's happening in our muscles. Now, in our muscles, there's bundles of these actin filaments in a highly ordered structure with bundles of these myosin molecular motors in between causing this contraction. So every time we contract our muscles, there's lots of these little molecules that are causing the contraction. But I'm interested in cell movement of things like these white blood cells that I showed you at the beginning. And there, we don't see these highly ordered structures of the actin. This was the picture I showed you earlier of the actin here. We don't see the resolution very well in this image, but if you remember that electron micrograph image and the super resolution image I showed you, the network of actin filaments was disordered, it was all over the place, and we don't see that clear order. But we still get this contraction. Not in one dimension, but we can get the contraction isotropically, and sometimes it's more directed. So how do living cells move? They are soft, like a gel. They are active, which means out of equilibrium. What I mean by that is they're consuming energy. And with the energy they consume, they can do two things. The filaments themselves can grow longer, and in interacting with these little molecular motors, they can cause stresses such as contraction. So the classic picture of how cells move is a combination of polymerization at the front, so growing filaments pushing the front forwards by growing longer, and at the back you can get contraction using these kind of mechanism, and together that can generate motion. I say together, often the two mechanisms are happening together, but just one of these mechanisms in different situations is enough to generate movement, and depending on the environment that the cell is in, then it will use different combinations of these mechanisms in order to move. Now, I've talked so far about different length scales, and I've got down as far as the length scale of a filament like this. But let's go a little bit smaller again and think about the individual subunits. So each individual subunit here is a protein. Now, if an individual subunit is going to add onto the end to make this filament grow and push the front of the cell forwards, then where does this little subunit come from? I just said it arrives and adds on the end. Well, it must be moving around in order to arrive. So how's it moving around? Let's think about the movement of these individual little subunits, these individual proteins. How are they moving around inside the cell? So here, if this works. Okay, I've got little molecules. I've got my little proteins moving around. Now, that's happening continually, that animation only runs once, I'm afraid. But they're moving around, and we call this movement Brownian motion. Now, Robert Brown, who is the scientist after whom we call this motion, was a biologist and studied initially pollen. And he was looking at pollen under his microscope, and he saw that the pollen was jiggling around in some kind of dance. He said it was like the molecules were dancing around, and he thought that that was because these grains of pollen, these particles of pollen, were alive. So he said this is jiggling around due to life, animate motion, but he was a good biologist, he wanted to test this hypothesis. So he went to a museum and he got some dead pollen, and he looked at that under the microscope, and that moved around. Oh dear, maybe it's not dead enough. So he looked at various different plants, such that he was sure that this pollen was really dead, and it was still moving around. Okay, well, let's really test this. So he got particles of soot, and he ground up metal, and a whole series of little particles that were very clearly dead, as in they'd never been alive, and they moved in exactly the same way. Now, back in, I should say this slide is where is his brief account of the microscopical observations of the particles contained in the pollen of plants and on the general existence of active molecules in organic and inorganic bodies. Quite a long title for a scientific article. I like it because he uses this word active, which we use today in a different context, a different setting that I've just explained to you. But this is his article, and in which he describes these beautiful observations. But Robert Brown showed that it wasn't because they were alive that was in this motion, that he didn't understand that motion. And it actually took nearly 100 years before scientists fully understood what we call Brownian motion. And it was actually Einstein who contributed to this story to really understand how Brownian motion works. And it was actually Einstein's PhD thesis. We tend to think of Einstein as being famous for relativity, but he worked on Brownian motion as well. And what was tricky about this is that Brownian motion is similar to, but a little bit different from something we know better, which is diffusion. And diffusion is the jiggling around of water molecules, say, due to thermal energy. If they've got some heat energy, then they can move around. The hotter they are, the more they move around, and they're jiggling around. That's fine, but when you talk about particles that are bigger, of the size of pollen grains that Robert Brown was looking at, they are actually too big. That was the problem. They're too big to move around by just thermal motion. What's actually happening is all the little water molecules are hitting in and kicking these big particles. When I say big, these are all the micrometers, or smaller fractions of micrometer, but they're water molecules are many orders of magnitude smaller than that. So actually, a single water molecule hitting one of these things isn't going to budge it very far. it's not going to move it very far because it's much bigger. And on average, the directions of the water molecules colliding with these particles are in all directions, and so on average, it's going to go nowhere. So that was the problem that scientists were stumped by. And the solution is that actually, very occasionally, in something we call a rare event, lots of water molecules happen to hit at the same time in the same direction, and that's what gives the particles a kick and makes them move. And the reason why we see this motion on the timescales that we have accessible to us is that the collision rate of water molecules hitting these things is so high. I did a back of the envelope calculation and I got 10 to the 12 collisions per second. That's a lot. And so a rare event, when you've got 10 to 12 events per second, means that actually we see these rare events on sensible timescales. So that's actually where this motion comes from. So these proteins that are monomers of my filaments are moving around with Brownian motion. But let's delve one level even smaller and think about motions inside a protein. So this is a protein that I did quite a lot of work on in my PhD. This is two images overlaid on top of each other. If you look in the background, you see these blue squiggly lines. That's the protein. This green thing here is DNA, but we're looking end-on to it. This is called the lactopressor. It's a protein that binds to DNA. It's a genetic switch, switches genes on and off. So we've got this protein here, but overlaid on top of the protein is a physicist's drawing of the protein, my drawing of the protein actually. You see I've hugely simplified this protein to two plates with a bunch of springs in between. And this was to help us to understand the internal vibrations of this molecule. That due to this Brownian motion I've been discussing, not only is this whole protein able to move around, but it's able to move internally vibrate. So bits of it are moving with respect to other bits. So it's basically doing this, yep, and doing this, different modes. So this is brings me on to talking about randomness. There's two meanings for the word random. First of all, in everyday life, we talk about the word random by meaning purposelessness, by chance, something happened at random. But within the scientific context that I'm working in, the word random has very specific meanings. So what I'm talking about when I say something is random or disordered is that microscopic details are unpredictable. If something is random or stochastic, it means that the microscopic details are unpredictable, either in theory or in practice, but we can't predict them. But having said that, macroscopic processes may be predictable. So this picture here is just white noise, it's just random, but sometimes these apparently random processes, like the random motion of these proteins wiggling around, can actually lead to predictable behaviour on a larger length scale. So a few words about my current research, which is about predicting cell motion. So I've talked about some of the ingredients, the components to the cell system, done a reductionist approach, and now I'm going to build up again, and I'm going to show you some results of calculations. So this is the results of the calculation here. We put into the system this. It's a circle. No, actually we did it in a sphere, but this is a 2D image of the sphere. And inside that sphere we put basically a liquid crystal, lots of little arrows, and they're all basically pointing this way, left to right. But you see some of them are pointing a little bit upwards and a little bit downwards. So they're not totally all pointing in the same direction. There's a certain spread. And then we calculate from that what the velocity should be. And what we've got here is we've got these directions, we put in the directions, and we put in this contraction that I talked about, this stress that's caused by the molecular motors interacting with these filaments. And then we calculate this. And you see these beautiful vortices in the velocity. The velocity is moving forwards here and round backwards in this circle, and this way it goes round like that, like the vortices. Now this is not a computer simulation. This is calculating the mathematics by hand. My PhD student Carl Whitfield did these calculations by hand, which is great, and so we've got great predictive power, but it's a hugely simplified system. We fixed it to be a sphere and not deform. We did lots of simplifications to be able to get something out of it mathematically. And so this is what we end up with. We've analysed the motion. So with these vortices, what we get is a force pushing forwards here and forces pushing backwards here, and this allows the whole thing to move forwards with respect to the medium that it's inside. And this kind of motion is called a squirmer in the literature. And so we've got this kind of motion that we predicted from an extremely simple model. We want to make it a little bit more realistic, because I said we fixed it to be a sphere. Well, we know that cells aren't actually spheres. So in order to do better than this, we did a simulation. Oh yes, there we go. Aha, did you see that? It was a bit quick. It's now moving. And what's exciting about this simulation is that we didn't cheat, like I said in the previous one, by starting with the arrows all pointing mostly forwards. We started with the arrows pointing in all directions randomly. And then the system, what we call it symmetry breaking. It started with the directions all at random, and then this direction emerged from the system. So let me show you that again. So initially it doesn't move because the directions are all random, and then it finds a direction and off it moves. And the direction it moves in here is random because we're not directing it in a particular way, but it chooses a direction and off it goes. We can do better than that in the sense that we can... Oh, gone too many. There we go. Oh dear. There we go. So we can do better than that in the sense that we can analyse different kinds of motion. So this was the motion I showed you here, the motile state. We also have the stationary state where it doesn't go anywhere, and we also have a state where it's deforming into a sort of dumbbell shape that looks like it's dividing. And what we varied here in the simulation is various parameters. We've varied the activity, which is how contractile is the system, and we've varied the surface tension, which is around the edge of our model cell. So this is, again, very simple. It's not really a cell, but it's a computational model to try to understand this motion. So that's a flavour of the sorts of things I do in my research, taking what we know of the ingredients and putting them together into a mathematical model, and then seeing what behaviour emerges from those ingredients. So what I've done so far is I started with the macroscopic picture of the cell moving. I then did a reductionist approach looking at the individual components in that system, and then I put what I knew about those individual components together into a mathematical model to attempt to predict the behaviour that those sorts of ingredients will give. And so, as you can see, my simulation is a long way off this real life situation of the white blood cell chasing the bacterium, but we are bit by bit understanding better the mechanisms of how a system like this works. So I look at a system like that and I see it's beautiful, it's got purpose to it, it's got the job to do that it's doing extremely well, but it's made up of ingredients that are, and components that are moving around at random with Brownian motion. There's lots of disorder in the system with ferments pointing in all kinds of different directions, and there's energy being inputted into the system, and together, those come together to give us what I'm calling here the emergent behaviour of this moving cell and its system. And so I'd like to conclude here by saying that what I've talked about is an ordered purposeful systems can emerge from microscopic disorder. So we can have disorder and random motion, random positions at the microscopic level, but we can have emergent structures that come from that, and those emergent behaviours and structures are beautiful and ordered look order to us. And for me, as a Christian, this leads me to look at systems like that, and I'm amazed. I'm filled with awe, filled with wonder. Wonder in two senses. Wonder at, wow, isn't that an amazing system? But also wonder in that it fills my mind with loads more questions. How does that work? What happens about this? What that? And that's why I'm a scientist, seeking to answer some of those questions, ask and answer some of those questions. But as a Christian, it also leads me to worship the Creator God, who I believe made these intricate systems, and this beautiful balance of disorder and order and chaotic details leading to emergent, ordered and beautiful processes, leads me to thank God for these beautiful systems and the way that He has made life here on earth. So that's where I would like to finish, and I'm happy to answer questions.
Thank you, Rhoda. Some fascinating bits of physics interacting with biology. We've got 20 minutes or so for questions. Let's start over here, shall we?
I think all our speakers have talked about beauty, but why do we think things are beautiful?
That's a fascinating question, and I, like Dirac and many physicists before me, look at equations, and for me certain equations are beautiful, and I understand that for many people in the audience you don't understand that concept. How can an equation be beautiful? But for me, I can look at some equations and realise that they are beautiful. And there are many things that we have in life that we look at and say are beautiful. Art is something that we look at and say it's beautiful, but some people look at pieces of modern art and they don't like them and they say that's not beautiful, whereas somebody else looks at it and says that's beautiful. So there is obviously some variations in terms of what we see as beautiful and what we don't, but there's lots in common with people. Most people will look at a picture like that and say, oh, it's beautiful in some sense. Where does that come from is a philosophical question. As a scientist, I don't have an answer to that, but as a Christian, I would say it points something towards the fact that as creatures made in the image of God, we are creative and have an ability to appreciate aesthetics and beauty in a way that reflects the Creator God. And so we look at something that's in God's creation and we see it as beautiful. That is in no way a scientific answer to your question, but as a Christian, I think it has to be something to do with the fact that we're made in the image of God, that we can see these things as beautiful.
A mathematician sees them as beautiful, some of the non-mathematicians see them as very scary.
Thank you very much for your talk. This question is a little bit being devil's advocate. I hope you'll forgive that. If it's too tough, please feel free to defer it to the panel later on. So, I'm a computer scientist now, just to give you a background. I did physics as a first degree, and I was at Leeds University, so I understand the issues and things that you're talking about. But one of the things that's puzzled me is what appears to be the layered structure of the physical world, which you've been talking about. Might be slightly different in the living world. We're living, things that we were talking about earlier, but in terms of the physical structure of the world we observe. And I do get the impression, in this layering, you may have an ordered layer and a disordered layer. We have an ordered layer to the visible world, but disorder beneath it with Heisenberg and quantum theory and so on. But maybe underneath that, there's another ordered layer, which I think they're still looking for, if I'm correct, in unified fields that bring all these things together. And if I could use this phrase, the more we try and drill down into understanding the structure of the natural world, the more complex it appears to be. Or maybe there are many more hierarchies than are currently in our understanding of the world. So what I'm suggesting to you, being devil's advocate, I'm not convinced by your argument, because I think our current view of the kind of things you're talking about depends on where we happen to be in the hierarchy at this particular moment in time. And the total situation may be much more complex than that. So I wouldn't necessarily draw the conclusion that you've drawn from the evidence that I think we've still got major problems.
Okay, thank you. So, first of all, I'd like to point out that my conclusion that's on the slide here is not purely a scientific conclusion. When I talk about emergence and beauty and wonder, I'm straying far from the field of science into metaphysics and theology there. So I don't want to claim that the science I've been talking about directly leads to that conclusion, because I made a metaphysical jump there. But to address some of those really interesting issues that you've been talking about, when people first were talking about atoms, the word atom means indivisible. And I think the word atom that we still use today is a very helpful reminder for us to be humble about the science that we know today. We know today that an atom is very far from being indivisible. We know something about the components of the atom. And we know we've got electrons, protons, neutrons, but then we discovered that protons and neutrons are not fundamental, and they're made up of quarks. And the point you raise is that, well, how do we know that quarks are the fundamental building blocks? So we do have to recognise that what we know now is only partial, and I've been talking about in terms of even what we're aware of, I've been talking about a very small range of length scales. I've talked about different length scales, actually a very small range of length scales when you compare that to the quantum level and the level of the whole universe. There's many more orders of magnitude I've not discussed. But I'd like to address one point about the difference between biology and physics on this issue. One of the beautiful things about physics, which makes much of physics doable, is that we can make assumptions about a particular length scale that separates one length scale away from other length scales. So, for example, if I'm looking at something at the micrometer or the meter length scale, I can ignore quantum mechanics effects. Great, makes it a lot easier to do. And if I'm looking at the quantum mechanical level, I can ignore the effect of the pull of the gravitational field of the sun. That also is rather helpful. So we do physics by separating length scales and making assumptions, and that makes it a lot easier to do the physics we do. Biology is more complicated in the sense that the length scales that are involved in biology are not separable in the way that we do in physics. So I've been using the word emergence here, and within the scientific field, people are actually usually, and myself included, nervous about using the word emergence because it actually implies a lot more than science. But there is a clear intricacy and connection between length scales in biology. And that is fascinating in the sense that there is causality going both ways, causality going from the smaller length scale to a larger length scale and back down from the larger length scale to the smaller length scales. And the length scales are interacting in a way that we cannot ignore if we want to understand the biological systems. This makes biological systems more difficult than physics in this manner, but also more interesting. And so in that sense, it's quite a complicated thing. I hope that's done some justice to your question.
We might come back to that one later this evening. Thank you, Alexander.
Thank you. Yeah, I think that's a very important point that you just made about the length scale separation and the interwovenness. So my question goes in exactly the same direction in the conclusion. You say ordered purposeful systems emerge from microscopic disorder. That's what you think you have shown in your talk. I would say the actual conclusion, as far as I understood your talk, is that ordered systems can channel microscopic disorder in order to achieve purposes. Because you have not shown how the system emerges, but you have shown how when you have a system, you can channel microscopic disorder in order to achieve a purpose. What do you think about that philosophical argument, which just turns causality the way around, as you just said with the length scale separation, that causality actually comes top down and not bottom up? I think it would be a completely different research project if you say, I want to show how from microscopic disorder a purposeful system emerges. And that's something that you have not shown, I think, in your talk.
Okay, thank you. I do think that the causality goes in both directions. What I've attempted to show in here is if we take some individual components of the system and put them together, then together they, what emerges in my PhD students' computer simulations is a behaviour that we didn't put in the beginning. We put in certain ingredients and then we get this spontaneous symmetry breaking and this motion coming from that. However, I do think the causality does go in the other way, in the other direction as well, top down. I've been talking about a bottom-up description, we're attempting to talk about a bottom-up description, but I think we can also talk about top-down descriptions. And I think this is also important in linking what I've been talking about with previous speaker this morning in terms of DNA. I think there's a causation both ways in there as well. So he was talking about how we have the central dogma and the DNA code translating into proteins, but then the proteins themselves, and he did mention this, then affect the DNA and can turn genes on and off. And in that sense, we've got the top down as well, and it's more of a circle. So I have no way done justice to this subject. I've just touched the surface of these issues of causality and directions.
Laura, right at the back.
Thank you. Thank you. I'd like to ask you about the little video clip where you've got the white blood cell chasing the bacteria.
Yeah.
Is that just a matter of electronic or chemical repulsion or attraction, or is there any, I mean, is the bacteria running, knowingly or unknowingly, or is it just chemical or?
Okay, thank you for that question. There are chemical gradients that are in the system, and bacteria are known to be able to sense chemical gradients. We call it chemotaxis, which is, taxis is movement, chemo is chemicals, so movement in response to chemicals. So bacterium can swim either up or down a chemical gradient. So that means they can swim towards a food source or away from an antibiotic, for example. So there is likely to be some directed motion of the bacterium in the system, and similarly of the white blood cell, and some directed motion of that as well in terms of chemicals. But chemicals are, your question was a little bit more general than that, and you were asking, what is it basically? So yes, chemical gradients can direct motion, but also mechanical gradients can direct motion, and that's more recent research has been focusing on mechanical gradients. We call it durotaxis rather than chemotaxis, and cells that are crawling on substrates are able to follow a gradient of stiffness in that substrate, as well as being able to follow chemical gradients. So yes, there's multiple things that can guide cell motion, but cell motion can also be spontaneous. So if you take a bacterium and put it in a medium where there are no gradients in chemicals, then it will move around in random directions. But if you put a chemical gradient in there, then it will perform what we call biased random motion, such that it is more likely to go in the direction it wants to than away from that direction. So it still wiggles around, and if you look at it on a short time scale, it looks random. If you look at it on a longer time scale, it is actually going to where it wants to go to. So there's chemical gradients are involved, mechanical gradients are involved, a variety of different gradients, but also spontaneous motion, choosing a direction at random.
Can it only behave in a programmed manner then, depending on the environment? Does it have any choice at all? Can it dysfunction?
I'm not quite sure I understand your question. So there's two types of motion. If you put a gradient on, then you can direct the direction that it will go. So in that sense, it's programmed. But if you put no gradient on, then it will choose a direction. the choice of direction will be at random, in the sense that if you run the simulation or repeat the experiment enough times, you will get a normal distribution of directions chosen at random.
Thank you.
It's all very easy for us to put sort of human feelings onto these sorts of things and imagine that the macrophage is perceiving the bacterium in the way that we do, but this is perceiving by chemical gradients and we say mechanical gradients as well. The process you described is random. Is the outcome at all uncertain?
There are some uncertainties in the outcome. For example, this discussion about the direction it chooses. If there's no gradients in the environment, then the direction it chooses is random. But the fact that it chooses a direction and moves is not random in that sense. So there's elements of randomness and elements of non-randomness.
I think if I spied over the side of your slide, but I've been naughty, I saw another bacterium that wasn't moving. Is that?
Oh, it wasn't moving at all. Yeah. Okay. So this is on a surface, and if a bacterium sticks to the surface, then it's not going to be moving around because it's not in the fluid. So you do that. I would say that that's likely to be why that particular bacterium is not moving at all, or it could be a dead one.
Or it's not being chased.
Or it's not being chased. In fact, the way the bacteria move is that they move if they're happy, as in they've got enough food. We're being very personifying here. If they've got enough food and nutrients, they will move at random, unless something's stopping them, like they're stuck to a surface, or they don't have enough food and then they stop moving.
But an antibiotic will clear it out, so the outcome is known in advance.
Well, I wish that was true, but with antibiotic resistance being more and more of a problem, actually we can no longer rely on our antibiotics. Antibiotics are interesting in the sense that many of the antibiotics we use, and those that bacteria are becoming resistant to, we don't actually understand the science of how they work. So there are many antibiotics that we use happily in medicine, but we don't understand all the scientific details of how they work.
Don't tell my students that, because I lecture antibiotics.
You know some of it.
Question there, yeah.
Just a,
Yeah, okay, I'll take that one and then Jeff, since you've got the microphone.
just a little observation and a question. Do you use much qualitative analysis as opposed to quantitative analysis to get to the truth? Because I noticed in the research that numbers are always the top of the tree, if you like. Actually, in biology, they're not. You know, a lot of the things that we analyse in biology are qualitative. Like you've illustrated some of those points in your answers to the mathematical challenge, but I think that actually you gave a very good answer because you gave it in terms of understanding the whole picture, which, as the previous speaker said, was only part of the truth anyway. So,
Yes, thank you. I actually think that both quantitative and qualitative are extremely important, and both play a part in what I've been talking about here. And despite being a physicist, and therefore people expect me to be quantitative, many of the conclusions of my calculations are qualitative, in the sense, if I show you here, there's qualitative aspects to this graph as well as quantitative aspects. So I would describe a qualitative aspect. Does it move or does it not move? Actually, I'm really interested in the answer to that, which is a qualitative question. Does it move or not? The quantitative question is how fast does it move? But actually, putting numbers into this, we don't have very good estimates for the numbers we need to put in. At best, we have about an order of magnitude estimate from experiments measuring various different numbers we need to put into this. So the actual numbers that I get out, I don't really trust actually that much. So the how fast does it move, which is the quantitative question, in some senses is important, but in other senses the qualitative question, does it move or not, is also important. So I think both are important, and I think we can get too hung up on numbers, and we can also have the opposite problem of running away from quantitative, and I think the two are important.
So maybe Hillary or somebody can tell me not to ask this at this moment, but I just, maybe during the rest of our weekend, we can explore a little bit more this, I think, a very important point that you made, which is that probabilistic or random processes, things that where we cannot detect any order, they truly do seem to be disordered, nevertheless lead to ordered outcomes in certain ways. And I just wonder, at least the milieu where I am, I think people have a hard time understanding how, on the one hand, God can act in a providential way, and yet there can be these random processes. Those seem to be completely incompatible, immiscible, to use Stephen's analogy this morning. And I just wonder, and yet biologists, I think, recognize, and biological physicists better than biologists, and I'm a biophysicist, which means, well, when I was getting my PhD, just you'll appreciate this, we used to ask, what is a biophysicist? And well, biophysicists, when they talk to physicists, talk biology. When biophysicists talk to biologists, they talk physics. And when biophysicists talk to one another, they talk about the weather. So I know not to be dangerous, but I just wonder if, this seems to be an important point. I think, I don't want to go totally theological here. I think there's some deep questions about metaphysical randomness and all of that, but I just wonder how does what you talked about address some of this for the average person in the pew, let's say?
Yeah. Thank you. It's an area I'm fascinated in, and I'd love to talk at more depth during the rest of the weekend. I think there's one thing about this story that physicists find, physicists are more comfortable with it than biologists, because physicists have grown up with statistical mechanics. Now, when I was an undergraduate physicist, I started my undergraduate course assuming that quantum mechanics was clearly the best and coolest part of physics, and I didn't know what statistical mechanics was, but I was blown away by statistical mechanics when I did lectures on statistical mechanics, because statistical mechanics takes this random, unpredictable microscopic details and predicts macroscopic behaviour. So the classic example is the air in this room. The particles of the air in this room are all moving at random directions, colliding with each other. They're moving with different velocities and moving all over the place in a chaotic way that we cannot predict the details. We cannot predict where the exact path of a single air molecule in this room, because there are so many collisions happening and the details are unpredictable. However, we can predict what the temperature of this room is. And that is something that physicists have been doing for a long time, that we average over these details that we can't predict, and we can get a very predictable average result. So on average, we know the temperature of this room. But ask us what an individual air molecule is doing, how fast it's moving, what direction it's moving in, we have no idea. But when we average over all the air molecules in this room, we have a very good idea what the temperature is. And that's something that physicists have grown up with and are comfortable with. Whereas in biology, you don't have similar ways of analysing, going from details that are unpredictable to predictable behaviour in quite the same way. So I think in the future, looking for a statistical mechanics of biology, if you want to put it that way, is what would be rather interesting.
But I think the theological question that you were asking, Jeff, is that God knows, providentially, God knows the outcome. Does God know every step along the process along the way? Is that?
Well, I come across a lot of people who are not scientists who would say, God is not the author of random processes, so how could a random process be something that reflects a providential creator? And I think the larger point that's raised here is that, well, there are random processes that seem to be harnessed providentially in very profound ways here, so that thinking that those are inimitable, that there's no way that you can mesh those, I think is perhaps mistaken. There's a guy, Richard Colling, who wrote a book called Random Designer, for example. He's trying to get at this idea.
Yeah, and sorry, may I just respond to that? I am comfortable with the idea that God can use random processes to produce predictable outcomes. But those people who don't share that belief are in extremely good company, because famously Einstein said, God does not play dice. And Einstein was not comfortable with this idea of randomness, and that comment was talking about quantum mechanics and the unpredictable nature of quantum mechanics, and the fact that we can only calculate probabilities of things happening in quantum mechanics. And Einstein did not buy quantum mechanics. He didn't like it. He spent 20 years trying to prove it was wrong, and because he failed to prove it was wrong, we learned so much about quantum mechanics. But he said he said that God doesn't play dice, and so I think it's a very interesting theological question.
So our time is nearly up. We've talked about the science. I'm going to give the last question to somebody with some theological background.
Well, actually, it was just a comment to follow up on the question of randomness, and actually you drew it to our attention to it in your slides, where you talked about the difference between the sort of popular understanding of chance, I think the word was, or randomness, I can't remember which, and the more scientific one. And I do think that actually semantics does come into play when you're talking about that. And so a lot of the problems people have when they talk about not buying the idea of randomness or chances, they haven't really understood what you're talking about biologically when you're saying that. That they have this kind of sort of rather nihilistic view, if you like, of it. So, but that's something we can pick up tonight, perhaps, in the discussion.
Thank you. I'm going to draw this
I'll draw this session to a close now. We're going to have a break between now and